Hey guys, today we'll be learning about the staining techniques. So your second lab exercise has gram staining technique. Gram staining is a technique used to differentiate the gram positive bacteria from gram negative bacteria. As I always said, there are multiple staining techniques in microbiology diagnostic labs. The very first technique that anybody can use is a simple staining technique. Simple staining is you just use one basic stain to quickly take a look at the shape and size of the cells. But that will not give you much of an information. So that is the reason we need to go for differential staining technique. As I already explained in your chapter 3, I believe, differential staining technique uses two different kinds of stains. Okay? So you have one stain that is known as a primary stain that is used and added to all the cells. You wash it off, you then go with another stain, you use the decolorizer to wash the stain off, and then you go with the second stain to differentiate the gram positive cells from the gram negative cells. Okay, so I certainly suggest, I highly recommend, and you must watch all the videos that have been uploaded on Canvas for the second lab exercise. Apart from that, you also should pay close attention to the procedures. I will be showing the demonstration now of the gram staining technique, but it's really important for you guys to understand why each and every reagent is added in the procedure. Gram staining technique has two parts. The first part is to make the bacterial smear. So what exactly is making a bacterial smear? You take the solid culture. So this is the solid culture where we have Klebsiella pneumonia growing on TSA plates, or you can also take the liquid culture. So this is Serratia marcescens, I believe, growing in the tryptic soy broth. So you can either use solid culture or liquid culture and add those cells on these microscopic slides. Okay, so this procedure is known as making a bacterial smear and making this ready for your gram staining procedure. Okay, let's just light the Bunsen burner. We have the flame going and we are ready to make the bacterial smear. So today I'm going to show you how to make the bacterial smear using the solid culture as well as the liquid culture. I have gram positive bacteria, Staphylococcus aureus, on the plate here, and gram negative bacteria, Serratia marcescens, as a liquid form. Okay. The goal is to add enough cells on the slide, on the microscopic slide, so that we can stain it and look those cells under the microscope. Okay, but the most important thing, very careful when you're picking the cells. You don't want too many cells on your slide, but at the same time you don't want too little. Okay, so as always, you have the inoculating loop, flame sterilize it. Wait for it to cool down. And pick few cells, very few cells. You don't want to have too many cells, because having too many cells would make your microscopic slide too crowded and you won't get good gram staining pictures. Remember, the goal is to be able to identify or differentiate between the gram positive versus gram negative bacterial cells on the slide. Think about a patient coming to a physician's office with an infection. The most important thing is the physician has to find out whether the infection is from gram negative bacteria or from gram-positive bacteria. Only then the right antibiotic can be given to the patient. So the very first technique would be, once you streak the plate, once the microbiologist would streak the plate, the next technique would be the gram staining technique. Okay? Alright, so you have the bacteria here, and because we have the bacteria as a solid culture, what we need to do is add a loop full of water so that you can dissolve the cells nicely in the water. Okay? The water need not be sterile because anyway we'll be fixing the cells and we'll be killing the cells, so we are not really growing the cells, so we don't need to really worry about water being sterile. You can use the DI water right on your tables. Okay? So again, water, you don't want too much. I am making sure